All right, so it's 8. So let's get to it. So reminder that hopefully you don't need a reminder, but test 2, it's in we'll open up an hour from now. It'll be open between 9 and 11, so you do have some time after we're done with our hour of lecture to study a little bit more if you would like, or if you have any technical difficulties, you have some time to try and figure it out. If you do run into a problem, like any technical difficulties, please contact me like ASAP. So I can be aware of that and we can work forward working through that. But otherwise, we're going to do a little bit of lecture. I'll probably do the same thing as I did for test 1. And, like I cut it short at 8:50. And then if anyone has any questions for 10 minutes, we can just do an open Q&A for any last things you might have for test 2. But test 2 should look very similar to test 1. The majority of the questions are multiple choice, and then maybe like 4 or 5 short answer questions to go along with that. But yeah, let's continue on what we were talking about on Monday. And we started our carbohydrate section. So we've been looking at our monosaccharides, and we just looked at the ones that are important for the cells, which are the box ones here. Glucose being the most important one, where our energy comes from. Then we looked at what a stereoisomer is. That is just where you have the same number of carbons, but different chirality. Then we looked at epimers, which is more specific in that you only have one different chiral carbon. So remember, that's a stereoisomer, and that is an epimer. And the sugars we were looking at were all linear. However, inside the cell, when we think about sugars, we're going to think of them as cyclic. And this transition between a linear form and a cyclic form of a sugar, that's spontaneous. There's an equilibrium there where they can switch between cyclic and linear all the time. And the way that happens is that the last chiral carbon of a sugar chain and the carbonyl carbon are going to be involved in a cyclization reaction. So hopefully you remember your arrow pushing from organic chemistry. But what happens is that the oxygen of the last chiral carbon will go and attack the carbonyl carbon, and you'll form a cycle. This happens in both aldehydes, and it also happens in ketones. And it's always going to be the same, where that last chiral carbon's oxygen is going to attack the carbonyl carbon. And generally, you're either going to be making a 6-membered ring or a 5-member ring. A 6-member ring is a pyranose. A 5-member ring is a furanose. Right? And when we cyclize our sugars, the numbering of the carbons stay the same in the cyclized form, in the linear form. So if we look at the linear form of glucose, here we have an aldehyde. Our carbonyl carbon will be carbon number 1, and you just move down the chain. So 1, 2, 3, 4, 5, 6. If we go to the cyclized sugar, here's carbon 1, our former carbonyl carbon, and it's color coded. And then we just follow 1, 2, 3, 4, 5, 6. And you can see our last carbon here is actually not part of the ring because it's outside when we do our attack. So this oxygen is going to attack this carbon, and now our circle is complete. The CH2OH is not part of the cyclization, so it's hanging out on the outside. Same idea with our ketones. Here, the ketone is carbon number 2. So it goes 1, 2, 3, 4, 5, 6. And when we do our attack and we're forming our ring, our carbons involved in the ring are only 2, 3, 4, and 5. And so now we have 2 carbon chains that are outside the ring. But the numbering again remains the same. 1, then 2. So this is our former carbonyl carbon as color coded. Then 3, 4, 5, 6. And during this cyclization reaction, the former carbonyl carbon gets a special name. It is called the anomeric carbon. So the anomeric carbon is the former carbonyl carbon. And what direction that OH is pointing compared to carbon 6, we name our sugars differently based on that. So in our representation here, our OH from the carbonyl carbon is pointing in the up direction. Carbon 6 is also pointing up above the ring. If our carbonyl carbon and our carbon number 6 are both pointing in the same direction of the ring, we call that a beta sugar. That's a kind of bad beta sugar. The way I remember that is that beta is both. That is both a carbonyl carbon and carbon number 6 are both pointing in the same direction. So that's a beta sugar. While if they were pointing in opposite directions, so let me draw that really quick here. So if our CH2OH was pointing up, but on the carbonyl carbon, our OH was pointing down, we call that an alpha sugar. Right? So what you're looking to determine if a sugar is alpha or beta, look at the position of the OH from the carbonyl carbon, compare that with carbon 6. Be aware we do this for both purines and furans, both purinose 6-membered rings and furanose 5-membered rings, where we look at the OH here, it's on carbon 2, that's pointing up. The C2H-OH of carbon 6 is also pointing up. So here we have another beta sugar. Any questions about that idea? All right. And we don't have to draw here, so we can skip question 6. On the test, I'm never going to ask you to draw anything, but I will say when you go and review for test 3, even though we're skipping this in class, this might be a good way to just remind yourself how the cyclization event happens, how to make a cyclized ribose from a linear ribose. So I would recommend that you do try and do this so you understand how the cyclization reaction happens, but just be aware I'm not going to ask you like on a test to draw a cyclized sugar for me, because drawing on Blackboard tests, I can imagine would be a giant pain. So we talked about alpha sugars and beta sugars. And we also said that this transformation between a linear sugar right here and a cyclic sugar, that interconverts inside the cell. You'll form a ring and then you'll open back up. And what this means is that the alpha and beta sugars, that will also, they can also change inside the cell. What I mean by that is that, let's say you're glucose and you're a linear form, and then that cyclization event happens. Depending on where your carbonyl carbon is, because remember, this is a single bond. Single bonds are free to spin. So this bond is just spinning in place, right? And so this oxygen and this hydrogen, they're just rotating around, right? In real time, they're just rotating. And depending when you attack, you'll form either an alpha sugar or a beta sugar. So it's all based on chance. It's all based on percentages. When we look at this inside the cell, we see that the beta sugar is preferred over the alpha sugar. And that's most likely due to the sterics of the attack, I would imagine, and any repulsion from this carbonyl carbon. But it is possible to have both the beta and the alpha. So what can happen is that, you're linear, you do attack, and you form the beta, and then you just stop being a ring and you go back to the linear form. Again, that's all spontaneous. These two forms are in equilibrium. Once you go back to the linear form, you could do a cyclization again, and maybe this time you just form an alpha glucose. And then you stay in the alpha ring conformation a bit, and then you go back to linear. So be aware that for monomeric sugars, as pointed out by the double arrows here, these transitions are happening all the time. You can go from cyclic to linear, linear to cyclic, alpha to beta, freely inside the cell, no energy needed. And so what I want to do right now is give you a little practice on determining if sugars are alpha or beta. And so since this is just these questions have simple alpha or beta component to them, I have a poll for this. So what I want you to do is determine for A, B, C, and D, based on what I've said already this morning. Are we looking at an alpha sugar or a beta sugar for each one of these sugars? When you have your answers, put your answers in the poll. Give you a couple minutes to do this, and then we will discuss. do about 30 more seconds for this. So if you haven't put your answers in yet, please do so. If you're not sure, just go ahead and put in something for the poll. Feel free to make guesses at this point. All right, let me just save that poll here really quick. Okay, saved. And it looks like, you know, we're pretty comfortable with this idea. Oops, did not mean to push that, but I'll just go over it quickly. So the first thing you need to do is find the anomeric carbon. And here's a hint for that. The anomeric carbon is always the one attached to two oxygens. So every carbon I circled is attached to two oxygens. Here in all these images, the anomeric carbon is always on the right. Be aware that they're not always going to be drawn like that. You can draw this ring in any orientation. So don't fall into the trap of thinking, okay, the carbon in the right is always anomeric. That's not true. Look for the carbon that's connected to two oxygens. That's going to be your anomeric. But let's go over this. So the OH is pointing down, CH2OH pointing up, different directions, alpha. OH pointing up, CH2OH pointing up, same direction, beta. OH pointing up, OH, CH2 pointing up, so that's beta. Here, OH pointing down, CH2OH pointing up, different directions, alpha. So it should be alpha, beta, beta, alpha. Any questions about that information? All right, seems like we got a pretty good hold on it. And let's talk about sugar modifications now. And inside the cell, you're not just going to have sugars that aren't changed. The cell makes tons of changes to your sugars, some of which you already probably know. So at position 2, if you take an OH and replace that with a hot hydrogen, that's called deoxy. So that's a deoxy sugar. If you remove the OH from carbon 6, you make what's called fructose. You can, instead of a carbonyl, you can have a carboxylic acid. And if that's an aldehyde, you made an aldonic acid. So that word, aldonic, is basically just combining two different words here. Aldehyde plus carbonic acid. So you take the first word, alda, then onic acid, you make an aldonic acid. While instead, if you take carbon 6 and do that, you made a uronic acid. So here, this was glucose, the 6th carbon made a carboxylic acid, glucouronic acid, uronic acid. If you take one of your sugars and add an NH2 instead of an OH, that's called an amino sugar. And they all have fancy names like 2-amino-2-deoxy alpha D glucopyranose. I would never expect you to be able to get that. It's not that hard, to be honest, but I'm more concerned about if I give you a sugar and it's missing an OH, do you know that's called a deoxy sugar? If I give you a sugar and one of the groups is an NH2, do you know that's called an amino sugar? And do you know the difference between a gluconic or an aldonic acid rather, and a uronic acid? And that's just the position of a carboxylic acid here. The sugars I'm showing here, ribitol, xylitol, glycerol, inositol, you don't need to memorize these or anything. These are just common sugars that have been modified, and they've been modified by having an oxygen there, or sorry, an alcohol there instead of a carbonyl carbon. These are called alditols, right? So if I take this carbonyl carbon and make an OH, that's an alditol. And if you've ever wondered how sugarless gum tastes so sweet without having sugar, and sugar is just glucose, is because it uses these alditols. It uses xylitol, and xylitol is very sweet to us, where you just need, I forget exactly like the magnification, but it's like 1,000 times sweeter than glucose to our tongues. And so you just add a little bit of xylitol and you now have gum that's very sweet, and you can say it's sugar-free because it doesn't have glucose. If you want to be specific about it, it's actually not sugar-free because xylitol is a carbohydrate, therefore it's a sugar. But how people usually use sugar, they mean glucose or fructose. So yeah, technically it is sugar-free. So yeah, those are the different modifications I would like you to be aware of. A deoxy, aldonic, uronic, alditol, or an amino. Not so much specific names of sugars, but just to recognize these modifications and what class they fall under. So any questions about that idea? All right. Questions, this is in my way. Question 8 is just asking you to draw some sugars. Again, for the sake of time, we're going to skip this. I'm not going to ask you to draw anything. A thiol sugar is, as it says, a CS bond. And so this is really a good way to test yourself of, do you know what a deoxy is? Do you know what the 2 position is? Do you know what a beta sugar is? So I would say, again, this is when you're going back for test 3, draw these to make sure you understand the naming system, the modification system. But on test 3, I'm not going to have you draw anything. And just for the sake of time today, we're just going to skip our drawing of the sugars so we can get on to more important things. And that more important thing is the idea of a glycosidic bond. Now, we already actually talked about glycosidic bonds when we talked about nucleotides, but when I use that word, it's probably one of those words that you heard and like it didn't have any meaning for you, so it kind of went in one ear and out the other. But now we can actually talk about what a glycosidic bond means. And a glycosidic bond is when you have a bond from your anomeric carbon connected to something else. So here in our nucleotides, what connects our sugar to our base is a glycosidic bond, because this was our anomeric carbon. We just lost the oxygen, but we're connected to the anomeric carbon to the base. And you have 2 types of anomeric carbons you can make. an O-linked, or an N-linked. And these just mean what atom are you connected to at the end of your glycosidic bond. So for nucleotides, we're always connected to a nitrogen on our base. So this is called an N-glycosidic bond. However, if we were connected to an oxygen, as I'm just kind of drawing here, that would be an O-linked glycosidic bond, because the atom at the end of our glycosidic bond is an oxygen. And like I have here in the notes, these are kind of similar to peptide bonds in our amino acids and our proteins, in that they're stable, hard to break. And glycosidic bonds are usually how sugars are attached to each other. And when you attach two sugars together, you get a disaccharide. If it's three, trisaccharide. And you can go on all the way to polysaccharide. Which is similar to our wording where we had a polypeptide. So polypeptide was for amino acids, proteins. Polysaccharide is if you have a bunch of sugars connected together. And you have two types of glycosidic bonds that can form between sugars. If you have your glycosidic bond between anomeric carbons, that's called non-reducing. So let me just draw that really quick here. So here, I'm not going to draw in all the OHs, but here was our anomeric carbon. Here was our anomeric carbon. They're connected to each other via a glycosidic bond. So we call that a non-reducing sugar. However, there, H here. However, if you have one anomeric carbon that is not part of a glycosidic bond, we call that a reducing sugar. So if you have an anomeric carbon that's part of a polysaccharide and it's not in a glycosidic bond, that sugar is called reducing. If the anomeric carbons are part of a glycosidic bond, that is called non-reducing. And those names come from an experiment you do to see if a molecule is reduced or not. Basically, if the sugar can form its linear form again, it can reduce that molecule. If it can't, it doesn't reduce it. And what I mean by that is if your anomeric carbon is not in a glycosidic bond, it is free to adopt the linear form. If you are stuck in a glycosidic bond, your anomeric carbon, you cannot form the linear form anymore. You are forever in a cyclic form until you break your glycosidic bond. All right, so any questions about glycosidic bonds, alpha, beta, or anything up to this point? All right. So we're just going to do a quick exercise here, and I want you to determine of the 4 sugars I have, can these sugars be reduced or can they not be reduced? So what I'm asking is, are these sugars reducing or are they non-reducing? I do not have a poll for this. So take a couple minutes and determine if these sugars are reducing or non-reducing. And I'll do A for us in case you're still not 100% sure. So the first thing you need to do is find the anomeric carbons. And the anomeric carbon, again, is a carbon connected to 2 oxygens. So this carbon is connected to 2 oxygens. This carbon is connected to 2 oxygens. These are our anomeric carbons. If both anomeric carbons are in a glycosidic bond, which they are here, they are bonded together, this is a non-reducing sugar. So again, take a couple minutes, figure out B, C, and D. Determine if these are reducing or non-reducing sugars. And I'll be back to discuss in a few. All right, so hopefully you had some time to look that over. Make this a little more clear to yourself. Let's do B. Again, we're looking for the anomeric carbon. We're looking for the carbons that are connected to 2 oxygens. So it's right here, and it's right here. So this carbon on the right is not part of a glycosidic bond. Therefore, this disaccharide is reducing. Remember, if one anomeric carbon is free, it is a reducing sugar. C, here's our anomeric carbon. Here's our anomeric carbon, oxygen, oxygen. Both are in a glycosidic bond, so that's non-reducing. And then D, here's our anomeric carbon, and it's not in a glycosidic bond, therefore it is reducing. All monomers are reducing. If you're not in a bond, glycosidic bond, you are reducing sugar. Any questions about reducing sugars here? All right. I believe that's the end of the PowerPoint from Monday. So let me just get the PowerPoint from for today up, and we'll just cover a little bit of that. Then we'll go into any questions people might have. So share this. Get my controls back up. All right. And we're going to continue our discussion of disaccharides here. And we're going to go into how we name the bonds that connect the disaccharide. So disaccharides have normal names based on what sugars are connected to them. For example, lactose is galactose plus glucose. Sucrose is glucose and fructose. I'm not interested if you know that lactose is made out of these two sugars or if sucrose is made out of these two sugars. What I'm interested in for this class is if you could look at the connection that combines disaccharides and if you can properly name it. So let's look at how we name the bonds that connect disaccharides. So let's start with lactose. And this is called a beta 1,4 connection. Let's look at why this is. First, we have our anomeric carbon being part of a bond. If your anomeric carbon is involved in the bond that connects disaccharides, you have to tell me if that anomeric carbon is either alpha or beta. Here we're saying beta because the bond is showing that the oxygen was pointing up. So here the bond connected to the oxygen, that's pointing in the upward direction, which was saying this OH was originally pointing up as well. And then we compare that to the CH2OH, that's pointing up too. So they were both pointing in the same direction. So it's beta, so that's where the beta comes from. And we have to say what carbon is part of our bond. Well, this is carbon 1 based on the naming scheme we went over today. Then we name the other carbon that's part of this bond. So it's 1, 2, 3, 4. So the fourth carbon is part of this bond. So it's beta 1, 4. Now, something that I see confused all the time when I ask questions on this on the exam. You only use alpha and beta if your anomeric carbon is part of this bond. Carbon 4 is not anomeric. So we do not say this is alpha or beta, right? Because this is a reducing sugar. Right now, this glucose is in the beta position. There's no reason why this sugar could not reopen up to be linear. And then when it reforms, this could become an alpha glucose, right? So only give alpha and beta if your glycosidic bond is part of the bond that connects your disaccharides. Can't stress that enough because every single time I've asked this question on the exam, and I'm going to ask it on test 3, at least 50% of the students will put like, oh, this is beta 4. And that doesn't make sense. You only use alpha and beta for anomeric carbons, like we see down here. So sucrose is a non-reducing sugar. Both anomeric carbons are part of the glycosidic bond. So we have to give both of these anomeric carbons a designation. So first, let's look at glucose here. The bond is pointing down. The glycosidic bond is pointing down from glucose. And our CH2OH is pointing up, which means these were opposite directions. So that's an alpha sugar. And the anomeric carbon, sugar 1, so that's alpha 1. And now our other carbon, our other anomeric carbon, so this sugar is pointing down. or that bond rather, is pointing down. The CH2OH is also pointing down. They're both pointing in the same direction, so that's beta. And this is a 5-membered ring. So our anomeric carbon is actually carbon 2 in a 5-member ring. So this is an alpha 1 beta 2 connection. So any questions about naming the bond between disaccharides here? All right. So of course, I'm gonna give you practice. I have 5 sugars here. I want you to name the bond that connects all 5 sugars. So name the linkages. Also, tell me if these sugars are reducing or if they're non-reducing. So this is the last exercise we're going to do. So I'm going to give you a few minutes, work through this. If you have any questions while doing this, let me know. Otherwise, I'm going to give you time to work and I'll be back to discuss in a few minutes. All right, so hopefully you had a little bit of time to look at these and work on these. Let's go through these quickly, starting with A. The first thing I always do for these is just find the anomeric carbon. And I'm looking for the carbon that's connected to 2 oxygens. So right here, oxygen 1, oxygen 2, right here, oxygen 1, oxygen 2. So this is carbon 1. The bond is pointing down. The CH2OH, which is right here, is pointing up. So this is an alpha 1. And it's connected to carbon 1 is right here. So this is 1, 2, 3, 4. So this is carbon 4. This is an alpha 1, 4. Remember, in 6-membered rings, which we're looking at here, the anomeric carbon is always carbon number 1. Then you move away from the oxygen while numbering. So this is 1, 2, 3, 4, 5, 6. If I was doing the whole numbering. So this is an alpha 1, 4. B, anomeric right there, oxygen, oxygen, and right here, oxygen, oxygen. So we have an anomeric carbon, part of our glycosidic bond. And so this is pointing up, as you can see, kind of like, if you compare A and B, you get a good image of what a up bond looks like and a down bond looks like, or above the ring and below the ring, I guess I should say. So this is above the ring. The CH2OH is also above the ring. So this is a beta 1, because we're both in the same direction. And then count our carbons, 1, 2, 3, 4. This is beta 1, 4. So same numbering between A and B, just difference between alpha and beta. So for C, here is my anomeric carbon. Here is my anomeric carbon, oxygen, 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 oxygen. So here we're pointing up. Here we're pointing up as well. So we're in the same direction. So that's a beta 1. Then my anomeric carbon here is number 1. And I move away from the oxygen while numbering. So that's 1, then 2. So this would be a beta 1, 2 linkage. So for D, again, I'm having carbon number 1 or my anomeric be in a bond. Be aware that this isn't always the case. In these examples, every one I always start with anomeric carbon. It is possible to have something like a 2 be your first bond, or 3 or 4, or any carbon really. So don't fall into the trap of always thinking, oh, the sugar on the left is always anomeric, always carbon 1. If I ever give you a different example, you will mess that up. So it's always good to go through this process of finding the anomeric carbons like I'm doing here. So this is anomeric, pointing down, CH2OH pointing up. They're opposite directions, so that's an alpha sugar. Anomeric in a 6-membered ring, so that's alpha 1. Here we have a 5-member ring for the first time. Our anomeric is here, oxygen, oxygen. So another tip, the anomeric carbon is always going to be next to the oxygen in the ring. So you just have to figure out which one is it. Is it the one to the right or the one to the left? Again, you're looking for the one directly connected to a different oxygen. And here our numbering system is carbon number 1 is the carbon connected to the anomeric carbon. So that's 1, 2, 3, 4, 5, 6. So it's actually carbon 6 that is connected to us. So this is an alpha 1, 6 sugar. And then for E, our anomeric carbon is right here. Our anomeric carbon is right here. So remember, the anomeric carbon is the former carbonyl carbon. Here, my glycosidic bond, I'm showing you the sugar being linear. Like I said, that can happen. these sugars go from linear to cyclic all the time inside the cell. So here our glycosidic bond is pointing away from us, as by the skinny line. So this is pointing away from us. And here our CH2OH is pointing towards us. So hopefully you remember from organic chemistry that if you have a wedge, what that is saying is that it's coming out of the page towards your face. A dash means it's going away from you. And if you just have a line, that means it's on the same plane as the paper. Because remember, we're looking at 3D models, or we're trying to represent 3D sugars on a 2D piece of paper. So we need ways to show that 3D. Since the bond connecting that oxygen and the CH2OH are different, this is an alpha sugar. They're not both doing the same thing. They're doing different things. So that's an alpha sugar. Our glycosidic bond right here. So that's alpha 1. And for ketones, the sugar that's above the anomeric, that's carbon 1. So that's 1, 2, 3. So this is an alpha 1, 3. And these are all reducing sugars. Every one. Because you're only non-reducing if both your anomeric carbons are part of the glycosidic bond. These 5 examples, I never gave you that. So every single one of these sugars is reducing. Any questions about our reducing sugars here? All right, if not, any last minute questions people want to ask about test 2? So a question is, what is idioacetate and when is it used? Idioacetate is a molecule that is used to prevent the reforming of disulfide bonds. And a lot of the times you don't use it. A lot of the times you just use beta-mercaptoethanol, and beta-mercaptoethanol breaks disulfide bonds. And as long as beta-mercaptoethanol is around, you won't reform those. But if you're doing protein sequencing, a lot of times you'll add idioacetate to as a way to prevent those disulfide bonds from ever forming again, because that acetate will interact with the sulfur of the cysteine and form almost like a pseudo-methionine where you have a CH3 there, and prevents the disulfide bonds from reforming. But that's what idioacetate does. So when a protein folds, G, H, and S are all negative, and the study guide said S was positive. Yeah, so that looks like to be an error, because protein folding, delta G free energy is negative, delta H slightly negative. Here we are decreasing chaos, so delta S should also be negative as well. Yep, so that looks like an error. The whole term minus T delta S would be positive, but the delta S itself would be negative. Anything else? Can you explain the difference between damsel chloride and Edmond? Sure. So damsel chloride is only for the N-terminus. What you do there is that you add a reagent, and the N-terminus gets this damsel chloride molecule on it, and you break all the other peptide bonds. And so the N-terminus has fluorescence now, and the fluorescence change based on what amino acid is connected to it. But that's all it's used for. You can tell what the N-terminal amino acid is. That's it. Edmond is more sophisticated in that you can do it by only breaking one peptide bond at a time. So basically with Edmond, you put the reagent on whatever the N-terminus is, then you break the peptide bond between the N-terminus and the next amino acid that's connected to it. You get the fluorescence of what was at the N-terminus, and you wash that away, and then you repeat with the new N-terminus. So with Edmond, with Edmond, you do it amino acid by amino acid by amino acid. Damsel chloride, you can only do the N-terminus, then you broke your protein. So what is the thermodynamics of protein unfolding? All right, so if a protein is unfolding, that means it must be spontaneous. All right, so the delta G depends on the conditions. And I'm going to assume the conditions we're talking about are, they are good for unfolding, right? So you're making unfolding spontaneous at that point. Delta S then, you are increasing chaos. So delta S is positive. Delta H is probably very small. Somewhere between very small negative, 0, and very small positive. This is actually a debate about what the sign of delta H is for protein folding and unfolding. It's probably super tiny, because any bonds you break in the protein, you're going to form those with water. So we don't quite know exactly the sign. But if conditions for unfolding are good, that is, it will unfold spontaneously, delta G has to be negative. All right, so that is 9. So I'm going to, your test should be up right now. As a reminder, feel free to use notes, just don't post it online. And yeah, if you have questions or you have problems, shoot me an email. I'll have my email up and I'll respond to you when I can. Otherwise, good luck. And I'm gonna cancel this out now. Last question. Would it be conservative mutation if one amino acid is nonpolar and the other is positive? Same with polar and negative. No. If you went from a threonine, which is polar, to an aspartic acid, that is not conservative. Conservative is only negative with negative, positive, positive, polar with polar, nonpolar with nonpolar. Polar and charged amino acids are different characteristics. All right, so I'm going to close this out. Good luck, and I will see you next week. Take care, everybody.